Hello, Biology 11, and welcome back. So we're nearing the end of the semester. Had we been in the lab, in real lab, physical lab, at this point we would begin our dissections. Today we would have been dissecting the clam, the squid, a crayfish, and a grasshopper. Well, maybe not so much dissecting those last two, just looking at the external anatomy. All right, so appropriately, this one is called animal diversity. So the aim of this lab is to show you some of the scope of life in terms of animal life that exists. All right, so as we learned yesterday, all animals have a common ancestor. They can all trace them back to the first animal species, and those species can trace back their ancestors way back to the first cells, right? So all life on Earth, as far as we have seen, have all arisen from a single ancestor. There haven't been multiple types of life forms evolving or being created from scratch. All right, so what comes from that, though, is that we can classify the animals. We can place them into uh, levels of classification that can increase or decrease in exclusivity. And scientists use morphological uh, characteristics, physical characteristics, that is, to help figure out what two animals are related to one another. Now, and that was how biology was done for centuries, basically, until the advent of DNA sequencing. And now we're at that point where if you want to figure out an animal, you have the ability and the lab and the money, you can take a DNA sample, send it out, or do your own analysis on the DNA sample and compare it to an online resource of DNA samples, and you can figure out what species it is from there and what it's related to. All right, and that's actually a vein of research where people just are taking DNA samples from all sorts of life forms, organisms, and trying to figure out how closely related they are to other organisms uh, in throughout the world. Okay, so but still, even today, you can't just do that in your kitchen at home. So we still use morphological characteristics as much as we can. Turns out that they align pretty well with DNA, with the DNA data that researchers get. Interestingly enough, there are times when the DNA gives you a surprising answer. And the reason for that is because sometimes morphology can trick you. In other words, the morphology of two organisms may be very similar, but it turns out that one of them is looks similar to the other one because it's trying to mimic it, or it's trying to sorry, not trying to, but it's what happened was convergent evolution, where uh, the environment has designed both animals very similarly, and it turns out they're not closely related at all. Okay, and sometimes the opposite effect happens, where you have one where you have one species, but it turns out it's two different species. If you look at their DNA, and they're very similar, but they look identical, and people used to think that they were one species. Okay, and that again is because of environmental conditions forcing that species to look a certain way or to act a certain way. All right, so some anatomical features that the lab book shows you that are used for organizing life forms into these hierarchies of classification is, for example, what level of organization does it have? We're talking about germ layers. So in terms of germ layers, does it have 2 or 3 germ layers? That is, 2 or 3 tissue layers in the embryo, actually in the zygote before it becomes an embryo. And each one of those layers is responsible for a certain set of organs or organ systems. How many? Some animals have 2, some animals have 1. I mean, no particular distinct germ layers, and some vast majority of animals have 3. What kind of symmetry are we talking about? Right? Do you have radial? Do you have bilateral, or something else? What's the body plan? All right. Do you have a mouth and an anus, or just one mouth, one opening for both jobs, or do you not have any kind of body plan at all? And also, the term, does it have a body cavity? Is it, in other words, is it coelomic or acoelomic? And we'll discuss that. And is it segmented? Does it have repetition of body parts? All right, so this is the cladogram, or tree cladogram, that's shown in your lab book, right? So you have the ancestral protist, which is the ancestor of animals, and it's actually the ancestor of protists are probably the ancestors of were the ancestors of all eukaryotic organisms. That includes fungi, animals, and plants. All right. From the single-celled protists, you evolved multicellularity, and it's not until then that you have an animal, right? So what makes an animal an animal? Well, it responds to stimuli very quickly. It can move around. There's a whole bunch of things that you can think about. But remember, animals are highly varied. So if the next level here we have the invention or the beginnings of the use of true tissue or germ layers, right? So if it doesn't have any of these germ layers, that is, specific tissues in the embryo that give rise to certain body systems or body organs, then if you don't have that, then you don't have these tissue or germ layers. And the group that best represents that are the sponges, right? And they're interesting. They're also called the parazoa. They're called the parazoa because they're next to the animal. Zoa meaning animal, para next to, right? Parazoa next to the animal. So the rest, everything else, are I guess you can consider them the regular animals. Sponges are considered animals. They're just a little bit different from all the other ones. And then, what about symmetry? Okay, does it have bilateral symmetry? So remember, bilateral symmetry, for example, a flatworm. Bilateral symmetry means you have one plane of division that can produce 2 mirror images. Radial symmetry, like a pizza pie or a circle. And very few groups have radial symmetry, and the only one that we're going to talk about today are the cnidarians, the jellyfish, the anemones, corals, things like that. All right. So cnidaria are the ones with radial symmetry. Okay, then what about the body cavity? Do you have a body cavity? Are you a coelomate? A coelomic cavity is what you're talking about. Coelomic cavity. Does it have a coelomic cavity or not? If it doesn't, doesn't have a coelomic cavity, then we end up with the flatworms. Oh, that's this guy. Okay, flatworms. A lot of which are parasites, by the way. Okay, and if it does have a coelomic cavity, does it have a true coelom or is it a false coelom? And I'm not going to go into the difference between the two because it's beyond the scope of this course. But basically, it deals with how many germ layers you have. Do you have all three or do you have two? Okay, and if you have that false coelom, you're probably going to be a roundworm, which there's all sorts of examples like the vinegary or the pinworm or the ascaris or ascaris, heartworms, a lot of parasites. Not all of them are parasites, but a lot of parasites. And if you are a true coelomate, you have a true coelom. Okay, when you're an embryo and you're a little ball of cells, and that little ball of cells starts making openings for the digestive cavity, is it going to make the mouth opening first, or is it going to make the anal opening first? And as it turns out, if you're what we call a protostome, then you make your mouth opening first. And that includes a lot of the things that includes all the animals we're going to dissect today. Well, that we would have been dissecting today, that we're going to discuss today. The mollusks, the arthropods, and also the annelids, which includes the earthworms and leeches. Right? And it's nice to note that among the protostomes, segmentation arose, as well as jointed appendages. Right? So an earthworm does not have a jointed appendage. Right? It doesn't have joints that help for the movement of certain limbs, for example. Right? It's just one large soft-bodied animal. And if you're a true coelomate and you have you as an embryo, when you're just a little ball of cells, you start making the little openings for the digestive system, and the first opening that you make ends up being your anus, then you're a deuterostome, and that includes us and sea stars and their group, their kin. Right? Note that we also have segmentation and jointed appendages. All right, so what are the germ layers? They are a set of cells that develop early on in the embryo. All right, and you can have a possible of up to 3, depending on the species that you are. What group are you in? Are you a sponge that has none of them, or are you one of these other things like jellyfish that only has 2? Or are you like us and has 3 tissue layers? Okay, so in this slide, I described what the 3 tissue layers can do for you, but I'm not really going to cover that too much. So let's move on. Okay, then we have symmetry. With symmetry, are you completely asymmetrical, like a sponge? You cannot find a place on a sponge where you can cut it in half and get 2 mirror images. Not in every individual, not in every species. Okay, because the sponges don't have that symmetrical body form. On the other hand, if you were to find a jellyfish or one of the related kin, like this hydra in the picture, then yeah, you have radial symmetry. You have the ability to slice the animal in half in many different locations, well, in different directions is what I mean. You'll get to those two mirror images. Compare that to what we have, which is bilateral symmetry, and there's only one plane division that will produce those two mirror images, right? Right, right, ooh, yeah, right down the face, down through the chest and the belly, etc. If you were to try cutting it in half top to bottom, the top doesn't look like the bottom, the back doesn't look like the front. There's only one way that you can do proper symmetrical division. So note, no, the term cephalization. Okay, so animals that have cephalization are animals that have bilateral symmetry. And what I mean by cephalization is presence of, let's see, a head or head region. There is a difference between having a head and having a region where your head would be. All right, so cephalization results in bilateral symmetry. So once you place your sense organs around your mouth, and that starts to create a head, you've lost the ability to be radially symmetrical. Because you have your brain, you have your auditory senses, you have your sense of smells, sense of vision up by near your mouth, then that pretty much you can't be divided in more than one way and produce mirror images. Then, of course, there's the body plan that we were discussing before. Well, what do you have? Do you have a body plan that looks like a sack, like a bag? Here's a bag, and that means there's no opening over here. There's only one opening, and whatever food has to go out the same way. So the mouth is used for feeding and excretion. Or is it a tube with a tube plan? Okay, so here's an example. Here's a sac body plan. Here, the radially symmetrical animals also have that sac body plan. Talking about jellyfish, corals, anemones. Okay, and no, there's no body opening over here. This is the only opening available. Whatever food to excrete the waste in the same direction, from the same direction. Tube within a tube body plan, the best example is an earthworm, right? Or just any kind of worm, really. You have a mouth opening, you have an anal opening on the other end, and a digestive tube that connects the two openings. Outside of the digestive tube, but still within the body, is a coelomic space. Okay, so this portion here, area in blue, kind of green in other places, is coelomic space. Right? And then there's the skin on the outside. So the skin and the coelomic space forms the first tube, and then the digestive tract forms that second tube. So you have a tube within a tube. Okay. We are not that different, right? You have an opening here, the anal opening there, and the digestive tract coils about, but let's just keep it simple, right? And there happens to also be a cavity, right, in the body, which is the thoracic cavity, the abdominal cavity. You know, your insides are not solid mass. There's a lot of space in there. The stomach and the lungs and the heart and the spleen and all your inner organs, they're all hanging out within a space inside your body that's outside of your digestive tract. So that space is the coelomic space. So we are also a tube within a tube body plan. All right. So this coelom, is often fluid filled, and I say here, it's lined by a mesoderm. Okay, so what does that mean? What is a mesoderm? Okay, the three tissue layers are ectoderm, mesoderm, endoderm. So the coelomic space is only created when the animal has a mesoderm. Right, and mesoderm allows for the development of various internal organs. Okay. So, does it have these spaces or not? If it doesn't have any of these spaces, we call it an acoelomate. A lot of times we have these flatworms. This is a flatworm. Extremely flat animal, right? And doesn't have any internal spaces. And definitely doesn't have any internal organs that would hang out in such spaces. So just remember, what does a coelom mean? And there are animals that have it, and there are animals that don't have it. Segmentation. All right, a division of some bodies into a series of semi-repetitive segments, a repetition of body parts. And you might say, well, that chart said that humans have segmentation. But last I checked, I don't remember being segmented like this, right? Like the centipede. Well, have you checked out your backbone? All right. Look, vertebrae, etc. Yes, each one is specialized and each vertebrae is a little bit different from the others, but basically it's a repetition of the same thing. All right. And you have your head, you have your thorax, and you have your abdomen. Your body's been segmented into 3 sections. Segmentation is much more easily observed in things like arthropods, like the centipede, where it has. Okay, what is that? All right, in the centipede, where it has easily visible segments. Okay, for example, this is one. This is another segment. Here's another, and there, and so on. Okay. Note, by the way, there's only one pair of legs per segment. Okay. One here, one there for this segment. Keep that in mind for a future slide for centipedes. All right, so using the information that we can gather from these morphological details, we can place animals in a hierarchical system of classification that, at the very top of this hierarchy, you have, um, well, let's say on one end of the hierarchy, you have a very inclusive group. So you can say, well, what domain is it in? Is it a bacteria, or is it an archaea, or is it a eukaryote? And then, everything falls into one of those three groups. And on the other end, you have something that's extremely exclusive, and it's so exclusive that it only contains one species. For example, here you have your system of classification. You have kingdom, phylum, class, order, family, genus, species. King Philip's class out for good spaghetti. Or whatever S word you want to put in. All right, that's easy to remember. King Philip's class out for good spaghetti. All right. And right above this is domain also. Domain Eukarya. Right, so this little bird is in the same domain as Euglena, or a paramecium, or amoeba, or plants, or fungus. We know that birds are animals. We also know from the fact that they have a backbone, and they have a dorsal on this side, a nerve cord, and other things, that they are chordates. Who else are chordates? Where we are chordates. Reptiles, other reptiles are chordates. Fish are chordates, for example. Birds are different from other chordates in that they have feathers, right? And they have a lot of other types of changes in their morphology, differences in their morphology of their bones, for example, or their brains and their breathing apparatus, that puts them in this classification, this class, all on their own. There are many different kinds of birds. There are hawks, there are seabirds like ducks, and there are songbirds, which are Passeriformes. So, okay, but which kind of Passeriforme is it? What kind of songbird is it? Well, there's a whole bunch of different kinds of songbirds. You have finches, and you have um, blackbirds, and you have warblers. That's the Parulidae. And this specific warbler is in the genus Cardellina and species versicolor. Right, so it's Cardellina. Its species name is Cardellina versicolor. Ooh, let's write it over here, nice and well. Cardellina. You don't have to know this name, of course. I'm just using it as an example. Species names are binomial, right? Two names. So that's the part you need to know. Species names are always binomial, as a genus and a species. The versicolor itself, by itself, has no meaning. It has to go in conjunction with Cardellina, and then you know what you're talking about. And the thing about this classification system is it's a lot like the metric system, in that if you say Cardellina versicolor to somebody in Australia, they know exactly, or they're able to find out exactly what you're talking about. But if you said pink-headed warbler, well, there are other types of warblers outside of the Americas that are no relation to this, and maybe they have a pink-headed warbler in some town in Asia, that the people around there call it a pink-headed warbler. Who knows, right? And then you're talking about 2 or 3 or 4 possible species, right? So scientific names, that's what this is, scientific name, are specific to one particular organism, and they're not used twice for different organisms, unlike common names, which can be used multiple times. All right. So we dive in now to the actual groups that we're going to be covering in this lab. The first group is phylum Mollusca, and that means these are the soft-bodied, mussel-like animals. Okay, and the phylum is very varied, and it includes things from cephalopods like the octopus and the nautilus, your octopus and nautilus, to slugs and snails and clams and oysters. Okay, and there are a few things that they all have in common with each other. Right. Well, they're all coelomic. You can find that out in that chart that they gave you in the beginning of the chapter. And what covers their coelomic space is something called the mantle. All right, so the mantle is an organ, and it's covering this coelomic space and covers the bodies of these organisms. And in those mollusks that produce shells, it's the mantle that makes the shell. So you have the shell, and then you have a mantle, and then you have the rest of the animal, going from the outside towards the inside. So they have a mantle. Their mouth is composed of a radula, which can be varied. And it could be something as simple as something that scrapes food off the rock, a scraping mouth, or it can be a sharp mouth that can tear at flesh. They all have a muscular foot, so that's a strong muscular appendage. All right, and their bodies, their organs, are bunched up into what we call a visceral mass. Okay, and they're all bilaterally symmetrical. Oops. Yeah, this is the next slide here. Okay, so there are 4 groups of mollusks, and you'll need these groups to fill out the lab book. All right, so there are 4 major classes. This one is the Polyplacophorans, which it looks exactly like what it sounds like. Poly meaning many, placophora meaning shells or plaques. Right, so here's one shell, here's another, there's another. This arrangement of hardened structures allows for a little flexibility in the animal. A muscular foot is found on the other side of this shell. It's attached. It's using it to attach itself to this coral, the pink structure around the animal, and the mouth is also located in there. And it uses its radula, the mouth, to scrape food off these rocks. Okay, so on the other hand, we have that second class, bivalves. This is an important one for you guys to remember. What is a bivalve? Examples. It sounds like what it looks like. What it sounds. Bi meaning two valves, right? By valves we mean shells. So we're talking about clams, oysters, scallops, So here's a blue mussel, and here's a clam. All right, all sorts of different. It's a very varied group. All sorts of different bivalves exist just in this area here in New York. Uh, and if you include the whole planet, it's a huge. There's a large number of biodiversity within this group alone. All right, so they also have a mouth on the inside. We're going to take a look at the inside of a clam in a few slides so you guys can see. So you have bivalves and polyplacophorans. Oh, by the way, a good common name for the polyplacophorans are the chitons, and these are good common names for the bivalves. All right, gastropods. They have their muscular foot, which is this. Attached to what looks to be their stomach, which is inside the shell. Right? So gastropod. Right? These are, as you can see, these are the snails and the slugs, and the big seashells, the conchs or conches, however you're going to say it. Right? And this is the first group that distinguishes itself by having cephalization. So here's the head region, right, with some sense organs. These little antennae of that. All right. However, even though this has cephalization, they all have bilateral symmetry. Okay, so slugs are also gastropods, right? You have sea slugs and your garden slug. That's extremely varied as well. And the last group are the cephalopods. The cephalopods are also extremely varied. All right, and they're interesting because they have a highly developed nervous system, which, in fact, the nerves of squid are the nerves that were used in the research that led us to an understanding of how nerves work. Right, so the first research that was done on the nervous system of anything was on the squid because the nerves were so big and easy to work with. Right. These animals happen to be very intelligent for the most part, especially some octopus. There's a PBS special on octopus that was, on Nature that was aired last year. If you can find it, I would recommend it. It's somebody who's keeping an octopus as a pet. They teach you all about the intelligence of these animals. And basically, these animals some octopus are smart enough to understand what's going on to them. They have a sense of self. They're self-aware, and I would ask you guys, don't eat octopus. All right, because it's kind of a little bit of a crime. All right. Okay. So before I said, the muscular foot is a characteristic that they all have, and so is the mantle. So here's the mantle, and these tentacles. These tentacles are the foot. Okay. Here are the tentacles there. Okay. Take a close look at the dissection of a squid. So we're going to take a closer look at this. All right. Before we go to share screen for a second. Let's go into the lab book. Okay. Okay, there we go. Okay, so here, the first feature of interest is formation of tissue layers. right? Here we go, tissue layers. Which phyla in the three only 2 tissue layers? Okay, which phyla? Okay, so remember that we have that hierarchy, and they're talking about phylum. This thing here. Right. So the major groups of animals are divided up into phyla. Which phyla tree have only 2 tissue layers? And that would be the cnidarians. And then another feature of interest is symmetry. Asymmetry means that the animal, okay, which phyla has radial symmetry? And that's again Cnidaria. The other have bilateral symmetry. Then they're talking about protostomes or deuterostomes. Which pattern of development do flatworms, rotifers, and nematodes? Here we go. Nematodes. Flatworms. And rotifers, which would probably in this group, in another group right around here. Rotifers. Okay, where would they lie? Well, they happen to be protostomes. Rotifers are very similar to arthropods, but not quite. Okay. Okay, so in this table, molluscan diversity, common name of a specimen. Well, let's say chitin, right? This is what I was referring to. Those are polyplacophorans. Polyplacophoran. You can say the description of the foot is under the shell. Under the shell is fine. Cephalization, no. Polyplacophorans do not have a particular head region. Then you had the oysters. Well, we know those are bivalves. And description of the foot, for an oyster, it would be something that looks like a tongue sticking out of the shell. Or the clam, right? Here's a clam. Okay, here's a clam. And maybe you've noticed a little thing, muscular foot right there, that sticks out, and it uses it to move around. And again, head region, no, not present. All right, and sea slug. What group are the sea slugs in? Do they have? How would you describe that muscular foot? Do they have a head region? And what about a nice vampire squid? Okay, or Humboldt, or whatever, some sort of octopus. What's the name of the group? And what would we call the foot? I told you what the foot was. Cephalization? Well, let's take a look. All right, and you guys can fill that out. Okay. All right, now determine the clam's dorsal and ventral regions. The valves are hinged. What is the function of a heavy shell? It's clearly for protection. Okay. All right. What is a mantle? Well, mantle is a body covering. Okay, in this case, for the clam produces the shell. All right, at this point we have to go back and take a look. So, with the dissection of the clam, we would have to identify a few things. Identify position of directions in the body. What is the interior end? Remember, the interior end is something that is close to the posterior end is something that's close to the anus. The ventral side, something that's close to the, like you can think of it on your body. Your ventral side is your tummy and chest, and the dorsal side is your back. They are not interchangeable or interchangeable terms. Anterior does not mean ventral. Posterior does not mean dorsal. They're separate. Okay, the adductor muscle is the first thing you're probably going to see. All right, you got to make sure you identify that from the pictures. And the purpose of that is that it closes and opens the shells of the clams. And identifying the foot, the muscular foot, we'll be able to see the gills, the mantle, and the siphons, even from just these pictures. Okay. So anterior versus posterior is a little bit tricky with clams, but here's the key. The umbo, let's change the color here. The umbo, little hinge where the two shells connect, always points in the direction of the interior side. Okay? So if you remember that, you'll know which way is interior, which way is posterior. The other end is, of course, posterior. And the hinge side is also the dorsal side. And, of course, the opposite would be the ventral. Right? Somewhere on this end is an opening for the mouth, which takes you to a digestive system, but we don't have to really know that. What I want you guys to be able to recognize is tissue that is attached shell. Here it is. Right? And everything else is on top of it. This is the mantle. There are some siphons here created by the mantle. There's an in-current siphon, and there's another one here, X-current siphon. Water goes in this direction, and water comes out in that siphon. Why does it need a siphon? Well, actually, most mollusks have a siphon, but this particular species, or this group, this class of mollusks, need siphons because they're filter feeders. So they take water in, water passes over this organ, this striated organ here. Remember, one of the ways to identify this is that it has little striations, little lines on it. These are the gills, also known as ctenidia. All right, they're a little bit sticky, and they extract oxygen from the water, and the stickiness of them extracts food particles from the water. So they're filtering the water of particles. Some of the healthiest bays or aquatic ecosystems have lots and lots of mollusks, specifically bivalves like these, in that ecosystem because they're constantly cleaning the water. In fact, one of the interesting things that I found that happened after Hurricane Sandy, I forget how long ago that was that clam hunters, people who go out clamming, they go out to the beaches and they collect clams. were not allowed to do so after the hurricane, because when the hurricane or any very strong storm hits, the sewage systems are overwhelmed in our cities, in our towns. And so all that extra sewage that doesn't get processed gets run off, gets taken out to the ocean. It runs off into the ocean and the other water systems. So what they were saying was that these clams that were out on the beaches were filtering the water that was contaminated with sewage. So it was not a good idea to collect these clams for consumption, for eating, right? Because these guys are filter feeders. Now here's that muscular foot. Let me change the color. Here's that muscular foot. Okay, that's the muscular foot that it uses. It sticks it out, push itself among when it's living in the sand. It uses that to push itself around. It's very thick. It's very strong. It doesn't have any internal organs on that. But then you have this structure, which is the visceral mass, which has all the internal organs. Note also the adductor muscles. Okay, you have posterior and anterior. Don't worry about this one, just consider it part of the posterior. So we have this and this. Two adductor muscles, one on each side. If you want to open a living clam, you're going to have to cut those muscles. Somehow sneak a knife in through between the shells and cut the muscle so that the muscles can't hold the shells closed anymore. So here's a picture of a real one, right? Here you have your abductor muscle. Let's change the color. Abductor muscle. Here's another. Okay. Abductor muscle, these are together. You have the gills. And here's your muscular foot. And this line, this is pointing at the mantle. So that's it, really. There are some siphons on this end, but we can't see them from here, so don't worry about it. What I really want you guys to be able to identify are these adductor muscles, the gills. Nope. Look at the gills. They have little lines in them, right? You should be able to recognize the mantle. It's this very thin tissue layer that's attached to the shell. What does the mantle do? It produces a shell, and it protects the body, right? And the muscular foot. Okay. Okay, so this slide I already spoke about. This is describing anterior, posterior, ventral, dorsal. All right, so here we have it again, dorsal. It's close to the hinge. The umbo is pointing at the interior end. You have the ventral end and the posterior. Okay, so let's do the dissection of the squid. And I'm going to ask you, of course, again, to identify dorsal, ventral, anterior, and posterior ends. All right. So during the final lab exam, there are going to be pictures of these things, and they're going to be unlabeled, and I'm going to ask you to identify them. All right, and you have to identify, again, the mantle, the tentacles, which is the muscular foot, beak, siphons, gonads, ink sac, a couple of things, but not too hard. Okay. Maybe the cecum. All right. So here is a diagram of one. Right on the top are the siphons. Right, and it's like a jet. Squid have like a jet propulsion system. They have this very muscular siphon, this funnel, and they can squeeze it very hard and shoot water out of the body with a lot of force. The animal is propelled in this direction while the water goes out in that direction. The tentacles are composed of short arms and long arms. You have long hunting tentacles, and the other ones, the short ones, are for grabbing. Inside that is the radula, which is, in terms of squid and octopus, happens to be a very muscular structure with a sharp beak. All right. And what we'll be able to see, maybe, we'll be able to see the ink sac. We'll be able to see the esophagus attached to the mouth. We'll be able to see the gills. I'm hoping I can show you the cecum / stomach and the gonads, because I'm going to show you pictures of a female that's full of eggs. All right, so know these parts. You have, and this the siphon, right? So this thing here is the siphon. Siphon, gills. Let's not call it a funnel. Let's call it a siphon. Gills, ink sac, cecum, esophagus, radula, which is the mouth. The arms / tentacles. Okay. So let's take a look at that. And I linked you to the website that I'm using for pictures. The link is on the PowerPoint. There it is, virtual squid. All right, so if we were in lab, you'd see your squid vacuum packed into these little bags like this. We put them on a dissecting pan. And the first thing you'll note is it has little fins. Oh, I can't write on this. Oh, I have my mouse. Okay, we have little fins. And this structure here is the mantle. And inside the mantle is the visceral mass, where you find the gills and the stomach and the heart and all that. By the way, these squid and octopus have what we call a closed circulatory system. They have blood vessels that connect to each other and to the heart. While in the clam, I think this is a question in your book, in the clam, the circulatory system is open. So they have a heart and a couple of blood vessels, but the blood vessels don't create a circuit. They don't connect to each other. All right. So here we have the head of the squid. This structure here with a little slit there, that's the funnel of the siphon. Here is the mantle. This is an eye. Remember I told you the squid have very well-developed nervous systems, and that includes the eye. Eye is very large, has a large lens, and their eyesight is superb. They can see just as well underwater as we can see out above the water. Right? You try seeing far away in underwater. We can't. Our eyes are not that good. Okay, so here are the two long tentacles and the 8 arms. 1, 2, 3, 4, 5, 6, 7, and 8. Actually, 1, 2, 3, 4, 5, 6, 7, 8. Okay. And this is 9 and 10. All right, so remember the muscular foot. So if I show you a picture like this on the exam and I point at this, at the tentacles, and I ask you, what is this? You should be able to say muscular foot. All right. If I point at this, you should be able to say, if it's this, you should be able to say siphon. All right. And if we were to looking at it closely, we'd see that each tentacle has these little rows, these circular rows of teeth, these suckers. And the suckers have little pointy structures we call teeth. All right, that helps them hold on to prey. All right, here they're showing you the funnel or the siphon. Right, and when you open the tentacles up, deep in there is the mouth. And if you were to cut it, which I would have asked you to do so, you would end up taking a large, round, muscular structure like this out. That has this little thin tube attached to it. That's the esophagus. So here's the mouth. It's been cleared of the muscle. And that's the sharp beak. All right? That's a sharp beak, and that's very sharp. I would ask you guys to feel it with your thumbs lightly, otherwise you might cut yourself. And it's a very sharp structure that has the composition of a thick nail. And here's the esophagus. And then we would cut open the squid. We would cut through the mantle, which is a thick organ here, in this case. Compare that to what we saw with the clam, which was a thin tissue, right? We can almost see through it. It was so thin. And it was attached to the shell, which, that's why it doesn't really need to be thick. These guys don't have an external shell anymore. They've evolved past that. But they still do have a little bit of a remnant of a shell. Here, okay, let me zoom up. Here's the mantle, it's open. And in the inside, here's a cecum. These are the gonads. And if you look closely, they're little round structures. These are eggs. So we're looking at a female. Right? This structure here, which is paired with another one over here, are the gills. And this dark section here is the ink sac. Here's a much better picture of that. Here's a gill. And on the other side is another one. Right, you can see these guys are bilaterally symmetrical. This is the plane of division where I'm passing the arrow. This dark black thing here, that's the ink sac. They use the ink to release the ink into the environment for protection. Okay. This is the little heart. Right. I don't like to ask about the heart. It's not that easily distinguishable, so don't worry about it. You have the gill again. This is a much better picture of the gill. And siphon over here, mantle. All right. Again, here's the ink sac. And here's the gonad, which has a different color than everything else. This is also probably a female and it's full of eggs. You remove the visceral mass, which is all these internal organs. Underneath, to the mantle, would be what we call the pen, and that is a thin shell. A very thin remnant of a shell from the ancestral condition. Okay. All right. So here's the shell again. This pen, we call it a pen. It's pointy at either end, and I guess it can give a little bit of a structural support for the mantle, but not much. Here they are removed. Other species have stronger pens than this. All right. So maybe we can answer a few questions from the book. Talking about the clam. The adductor, two Ds, right? The adductor muscles hold the valves together. After these muscles are cut, the valve can be carefully lifted. What is the advantage of having powerful adductor muscles? You can say protection. By keeping the shells closed. In fact, they're so good, very few things are able to open those. Sea stars are the only thing I can think of that have the strength to pry the two shells apart. Otherwise, other organisms, they've taken to drilling holes through the shells very slowly, but slow and steady wins the race. And like some gastropods called oyster drills, and they do exactly that. They drill little holes in the oysters until they reach the inside of the shell, and then they're able to take out the food. All right. Explain the term mantle cavity. All right, so I told you the mantle surrounds the body, right? So the space, they're talking about the space between the mantle and the organs. Between the mantle and the visceral mass and the muscular foot and the gills. Identify the incurrent and excurrent siphons. I showed you that on the diagram anyway. Explain how water enters and exits the mantle cavity. All right, so water enters via the incurrent siphon, exits via the excurrent. Expose the gills. Does the clam have a respiratory system? Of course it does. It just told you it has gills, right? What type of respiratory system? So the answer is yes, and gills. Why is the clam called a filter feeder? All right, go back to the to this presentation, which I hope I'm recording. Right. Yep, I am recording. Thank God. I've been traumatized by yesterday. Okay, so go back to the recording. If you didn't hear the answer, I explained that one. All right, and you can fill that in. A clam has an open circulatory system. Explain. I also explained that before. So make sure you're able to go back and use that information to fill this in. There's no point in me rewriting it after I said it. Here's a nice diagram of this animal. So remember, you don't need the internal organs. You know, your foot, the location of the mouth, because that tells you that it's the anterior. This portion points at the interior. Anterior adductor muscle. Here you have posterior adductor muscle. The mantle. Again, the mantle. Know your gills, incurrent and excurrent siphon location is good. Is good to know. All right, so that's it for that. Anatomy of the squid. Explain how both clams and squids are adapted to their way of life. Well, the squid are predators, right? With a muscular, talk about the muscular foot that has evolved into tentacles for grabbing prey. The clam have are filter feeders. And talk about, the clams are filter feeders. They have these gills that filter food out of the water. And talk about the gills and the muscular foot, which helps them move around slowly. Right? Clams don't need to move around too much. All right, feeding mode. Filter feeder. Filter feeder. Hunter. Let's call it a predator, right? Skeleton. External shell. Absent. I don't remember any skeletons in the squid. That little pen, that's not a skeleton. Circulation. Open. Closed. Cephalization. Absent. I've never seen a clam with a head. Squid. Present. Advanced. Locomotion. Muscular foot. For the squid. Jet propulsion. Propulsion. With the siphon. Nervous system. Minimal, I guess. Includes a brain. Eyes. This is advanced. Okay, here's the anatomy of the squid. Again, know your gills. Be able to identify the ink sac. Okay, so gills, the funnel, tentacles, / arms, radula, esophagus. I can show you a picture of the esophagus. Right, as it was attached to the head, to the mouth. You have that big muscular thing with the squiggly line, and it had that beak. All right, so this is the mouth, and that's the esophagus. All right, and the mantle. All right. Okay, let's take a 5-minute break here, and then we'll continue with the rest of the chapter. Okay, guys, I'm back. All right, here we have. We're going to continue now with the rest of the chapter, which is moving on to the arthropods. So, what are the arthropods? Well, the arthropods are, we already discussed, they're segmented, jointed animals, but they have a hardened outer skeleton, external skeleton. We call the exoskeleton. And it's because of this hard exoskeleton that they need these joints for the appendages so that they're able to move around. All arthropods must shed. Shed the exoskeleton. All right, so okay, so the hardened exoskeleton cannot grow with the animal. So as the animal needs to grow, it must shed its exoskeleton, and then it'll pump up its body size with fluids, and then it'll form a new exoskeleton. Right? So the result is that arthropods grow in stages. Right? They don't continuously grow every day; they just grow in stages. So they have a stage that they obtain as soon as they come out of the egg. Then they shed, and they have another stage, and then another stage after that, and so on, and so on. And they also have an open circulatory system, all of them. Right? And we're going to be covering 5 major subphylums. So remember, I spoke to you about what a phylum was. Well, subphylum, these classifications structures can be subdivided. And if class is too exclusive, but phylum is too inclusive, then you can add things in between. So, for example, we're going to add subphylum. All right, so that's a little bit. The subphylum is a little higher in the classification system than a class is, but lower than phylum. Okay, so the first group, subphylum Chelicerata, serrata or serrata, however you want to say it. These include, I don't know if you guys have seen them, the horseshoe crabs. Maybe you've seen them at the beach. They look like a large shell, like a large rounded crab. I should have included a picture of it, but anyway, they're sweethearts. They're huge, but they're sweethearts. You can put them on your face and let them crawl on you. I wouldn't do that because sometimes they have sharp little portions of the legs, and they don't mean to, but they might scratch you up. But they don't bite, they don't hurt. Okay. Anyway, they and arachnids, which include spiders, ticks, mites, and scorpions, they all belong to the subphylum Chelicerata. And that's because you see these little structures here. These little structures. These are the chelicerae. Chelicerae. I'm not going to hold you to that, though. I'm not going to hold you to this information, just in case you're curious. For the spiders, for all of them, they use these little structures for bringing food into their mouths. Right. So the fangs of a spider, for example, and scorpions too. Right. And keep in mind how many pairs of legs these things have. These guys have. The arachnids have 4 pairs of legs. The horseshoe crab has more, but since we're not talking about the horseshoe crabs, don't worry about it. So remember, arachnids have 4 pairs of legs. Subphylum Myriapoda, which includes centipedes and millipedes. All right, so these guys are segmented throughout, and these guys have two pairs of legs per segment. So the millipedes have two pairs of legs per segment, and here's a segment of the centipede. Here's another segment, and each one of these only has one pair of legs. So one pair per segment. Okay? One pair per segment. Subphylum Crustacea. The crustaceans, you know them as the shrimp, the crabs, the lobsters, the crayfish. We're going to be talking about crayfish today. And remember back to our first lab where we discussed the pillbug. The pillbug is a crustacean. It's a specific type of crustacean, but it's a crustacean. And these guys have 4 to 7 pairs of legs, right, depending on the group. Remember, they don't have wings, right? They're evolved for living in humid environments. Most of them live in aquatic environments. You definitely can't use wings in that. 2 pairs of antennae, and here are some examples, like crabs, pillbugs, shrimp, crayfish, lobsters. Crustaceans are particularly varied. So you have these larger crustaceans, and you have micro crustaceans, like the water fleas, Daphnia, that is, and the copepods, things like that you can explore in any of the ponds in our parks during the spring and summer months. And you have the hexapods. These are the insects. They have 3 main portions of the body, which is the head, thorax, and the abdomen. Head, thorax, and abdomen. And they have 3 pairs of legs. Here's one, here's 2, and 3. Okay? This is part of it here. This is the middle one. All right, most of them have wings, so some of the primitive insects are wingless. But, your best examples of insects would be butterflies, bees, ants, cockroaches, grasshoppers, termites. All right, all of these, beetles, all of those have wings. All right, so let's talk about the crayfish, though, because that's the first one that we would have been dissecting or looking at under the microscope. We would have been dissecting it a little bit. All right, we're going to be doing looking at the external anatomy and a little bit of the internal. So forget that. All right. I want you to be able to identify compound eyes, the antennae, uropods, cephalothorax. Remember, I showed you the 3 body portions of the insects. Well, crustaceans only have 2, right? The carapace, which is the external shell. We'll see the gills, the swimmerets. Okay. Yeah, I did not update this slide. Let's get rid of this. Well, let's take a look. All right. Well, here's a crayfish, right? This is the underside of a crayfish. Here's the top side. And we're going to first take a look at this portion here to be able to tell the difference between male and females. And that's this. These are the pictures for that. All right, here we have a male crayfish. And this is the male copulatory organ. So it's like a little leg, almost looks like a little leg. It's strong and rigid, and it's like these other structures that line the tail, which are the swimmerets, right? Because the tail continues down, right? They have little swimmerets that keep going, little appendages that help them swim around. And it's easier to tell the female because the female is missing that rigid structure. It's very short, flexible swimmerets. You know, see how floppy they are? And it has a little opening here, which is the seminal receptacle. It's what accepts the sperm packets from the male. Right. So, walking legs. 1. The claw is considered one of them. This one here. 2, 3, 4, 5. Okay. These fin-like structures at the end of the tail, these are considered the uropods. They're there for aid in locomotion. Right, and we're going to take a look at the gills next. These are the swimmerets that I was telling you about before. In the male, this one is very tough, rigid. The female, they're floppy, and the female would have this opening of the sperm duct. So make sure you are able to recognize that. Right, what is the carapace? The carapace is, of course, the shell that surrounds the top side of the body. So here's the carapace. And if we were to lift the carapace just above the legs, we would end up seeing this on the bottom picture. What are these things? These are the gills. They look like little feather dusters, or wet ones anyway. But they look like little feathers. They have an enormous surface area, so that when they come into contact with water, and how do they come into contact with water? The movement of the legs, right? So I forgot to include a picture, but basically the gills are attached to the legs, and as the legs move, water moves up into this right underneath the carapace and floods the gills, and the gills are able to extract oxygen in that way. Right. So here's the tail. And note the tail is highly segmented, right, which seems to be an ancestral or primitive characteristic. You find that in scorpions. You find it in, but you don't find it in spiders. Spiders are a little bit more advanced, more evolved, divergent is the word, than the scorpions, and you find that crustaceans have these segmented tails, those segmented abdomens, and in the insects, I'll talk to you about the insects next, about who has these segmented abdomens and who doesn't. Right. The last thing, so you have the abdomen here, and this section here is a combination of the head and the thorax, which we call the cephalothorax. So make sure you know that one. Cephalothorax. Look at this abdomen here. It's highly segmented as well. All right. It turns out that this is an insect that is, I guess you could say, I mean, I hate to say some animals are more advanced than others, but I would rather say this animal is not as divergent. In other words, it doesn't have specific characteristics that it has evolved that are different from the ancestor. Right, so this one's more similar to the ancestral condition. Right, like for example, yesterday I showed you the wing of the forelimb of a bird, or a bat, which were wings, and a little bit different from the ancestral forelimb, very similar to what we have, a very ancestral forelimb. In comparison to a bat or a bird, except that our fingers are great for grasping. Our hands are anyway, but the rest of the arm is very similar. But for the bats and the birds, the bones were a little bit different. Same kinds of bones, but a little bit different in how they were structured. So they were a little bit divergent from the ancestral condition. This grasshopper that's not divergent from the ancestral condition. Right, so this one is an insect, right? It has a head, a thorax, and an abdomen. Here it is, head, thorax, abdomen. They have wings, compound eye. I told you that you should look at the compound eye of the crayfish. Don't worry about it for the crayfish. We're going to take a close look at that for the insects. Of course, the antennae. It happens to have an ear here, which is the tympanum. And it's got 3 pairs of walking legs, all attached to the thorax. There are no walking legs attached to the abdomen, or the head, of course. All right, and we're going to distinguish the difference between males and females in a few. So here we have antennae, compound eye. It also has a little simple eye next to it. It's the tympanum. Forewing and hind wing, so it's 2 pairs of wings. 1, 2, and 3 sets of legs. Right? Know the head, thorax, abdomen. And how do these guys breathe? Well, they take in air through little holes along the abdomen, which are called spiracles, and they're attached to these tracheids, little tubes, canals that, with a little muscle power, are able to transport oxygen to the rest of the body. But it's not like us that we have lungs with a diaphragm that pushes air into our body. So here's a male versus females at the end of the grasshopper comparison pictures. A female will have these open clasper-like circus, but don't worry about that word. All right, what I really want you to remember is that this is called an ovipositor. And when the female is ready, a little long tube comes out, and the eggs will travel through that tube from this opening, and she'll insert the tube into a sandy substrate, sandy soil or something like that. The eggs will lay in there. The male does not, it still has a circus, but does not have that opening. So it's more of a rounder structure. Okay. Here are the spiracles again. Give you a close-up. This little hole there, that little hole there, and here, these are the spiracles. Actual openings for air. Now note that there's no organ that sucks air in. What that means is that arthropods in general, especially land arthropods, have a limit to the size of the body, because air can only diffuse, oxygen can only diffuse a certain distance before it's unable to reach any organ with efficiency. Right? So air is coming in through here, but it can travel as far as the head, and maybe it can travel a little bit farther. But at certain sizes, it's really hard for oxygen to saturate the whole body. So there is a limit to the size of land arthropods. The largest land arthropod that doesn't use gills uh, is like the bird-eating spider, which is the size of a human hand, of a large human hand. It's the biggest spider. And it doesn't, you can't get those giant insects that you see in the 1950s horror films, like They, or Them, or whatever it was called. The giant ants or the giant spiders. Okay. Those arthropods that use gills, that's a different story, though. Right? Gills are a little bit more efficient at capturing oxygen, and they can get a little bit large. But again, there's a limit to their size because of the weight of their shells. All right. Well, the spiracles. This story on the spiracles. What about the compound eye? Well, the compound eye is called the compound eye because it has multiple lenses. So each one of these little here's the eye. It's composed of something like this. These little hexagonal patterns. Each one of these little hexagons is a lens. And their nervous system has to figure out how to make sense of all of this, right? How to make sense of all these multiple images that are coming in from a single eye. Don't worry about them. They know how to do that. And so it's too bad we're not in the lab. It's really interesting to see this. To be able to take a look at this eye underneath the dissecting microscope. So, you've probably heard of metamorphosis. Speaking when learning about insects. Metamorphosis is a term that's used for a change of life. Amphibians, for example, have metamorphosis too. These are vertebrates, not arthropods at all. They change from a larval stage to an adult stage. This term is used for the exact same reason in arthropods, in insects. The only arthropods to undergo metamorphosis. Not all insects undergo metamorphosis. There are those that are not too divergent from the ancestral condition that don't undergo metamorphosis. So you can call that incomplete metamorphosis. And there are those that do have a drastic change. They go from something that looks like a worm, basically a larval stage, to something that looks completely different in the adult stage. Right. So, for example, the ancestral condition, these animals that have the ancestral condition have lots of segmentation on the abdomen, undergo incomplete metamorphosis. You have the adult that lays eggs. Out of the egg hatches a little miniature version of the adult. It grows by molting, right? Shedding the exoskeleton in stages. Here's one stage. So here's the first stage. Here's the second stage, third stage, fourth stage, fifth stage, and final sixth stage. They're born without wings, and eventually they develop wings. So that's incomplete metamorphosis. The young are called the nymphs. Dragonflies do this, right? Dragonflies, if you ever take a look at a picture of a dragonfly, also has this highly segmented abdomen. Now, what about complete metamorphosis? Complete metamorphosis includes things like ants, beetles, bees, wasps, moths, flies, and moths and butterflies. Start off with a I don't know, some of you probably know this by heart if you children or you've had the book, The Very Hungry Caterpillar, read to you. You know, in the light of the moon on a dark night, something like that, the moth lays an egg on a leaf, whatever, something like that, right? And out pops a little caterpillar. That's the larval stage. And the caterpillar is able to grow by shedding its exoskeleton as well. It's a little bit easier because the exoskeleton happens to be very thin, a very thin cuticle. And it has stages also. And the last stage is the large stage, and it prepares by entering the pupa stage. And we know this in butterflies as the chrysalis. And in moths, like this one that's pictured here, in moths, the caterpillar takes it another step, and surrounds the chrysalis in a cocoon. So maybe you were wondering, well, where does the cocoon come in the story? Well, that's where it is. Moths, not butterflies, moths will place the chrysalis inside a cocoon, which is a layer of leaves or silk or something else that surrounds the chrysalis, which is the pupa. Okay, flies will just have a pupa, and beetles and things like that. Ants, they all just have a little egg-like structure, which is a pupa. Okay. All right, so that ends the arthropods. I'm going to talk quickly about the chordates. Phylum Chordata, which includes the vertebrates. All right, so there are other chordates besides vertebrates. They all have a notochord. A notochord is a supporting rod. Here it is, supporting rod. And it extends from one side of the body to the other. A dorsal nerve cord is present in all of them. For us, we call it a spinal cord. Pharyngeal pouches. I showed you those pharyngeal pouches yesterday in yesterday's lab manual chapter. Right, pouches that are present on the throats of those embryos. Well, turns out those are the same things as gills for fish. Embryonic humans have them. Okay. And a post-anal tail, that is an extension of the body beyond the opening of the anus. Post-anal tail. Right? And you might be saying, well, I don't have it. I don't have that. We're humans, and humans don't have it. Well, you did when you were an embryo. And you should be familiar with the different classes of vertebrates. You have the sharks, which are the chondrichthyes. The bony, let's not learn this. Let's learn bony fish. It's osteoichthyes, that's bony fish, and these are the cartilaginous. All right. I think these scientific terms might be a little bit too much for Bio 11. All right. Cartilaginous fishes. Let me write that better. Okay. Cartilaginous. All right. So what that means is that sharks have a skeleton of cartilage, not made of bone. It's made of cartilage, like the material inside your ears or at the tip of your nose. Okay. The bony fish are different. Their skeleton's made of bone, which is a repository for calcium. And you have class amphibia, which are the amphibians. These are the frogs, toads, right, salamanders, and another group which I won't mention. I think it's too much. And you have class reptiles, reptilia, lizards, snakes, turtles, crocodiles, crocodilians, which nowadays also includes the birds. But let's learn class aves anyway. Let's learn it anyway, right? Aves, which is the birds. If you know Spanish, you're a leg up on this term, and or French maybe, any of the Latin languages. And the last class are the mammals. All right, and that would segue us into the section of a fetal pig for tomorrow. Not tomorrow, Monday, right? Oh, today's Thursday. Good. All right, so we'll stop there. Except I'm going to go over the lab manual now. All right, let me share that screen so we can see that. I almost forgot. So where were we? Arthropods. Arthropods are segmented like the annelids. These are the worms, earthworms. If you're wondering what an annelid is, earthworms, leeches is another example. But specialization of segments has occurred. Okay, so earthworms have specialized segments too, but arthropods take this to another level. So there's segmentation with specialization. Right. Segmentation. This is repetition of body parts with specialization. So each body part is now specialized for a certain function. Right. Each segment has a special function. Right, so the head segment of insects holds the sensory organs. The thorax holds the locomotive organs, like the legs or the feeding arms or whatever. Okay, so arthropod diversity. Common name of a specimen. Oh, a centipede. Okay, what was the name of the centipede? That's in the PowerPoint. Okay. Appendages. Remember, I mentioned how many appendages per segment. Don't say 100 legs. Centipedes don't have 100 legs. In this case, I'll give it to you anyway. 1 pair per segment. And the millipedes had 2 pairs. How about the spiders? Right. Okay, you can say arachnids, or you can say chelicerates, however you want to say it. We'll call it arachnids. Everybody knows that anyway. Arachnids. And how many pairs of legs do arachnids have? 4. How about crayfish, right? We talked about crayfish. What group are they in? And how many pairs of legs? We counted those. And let's say a moth. What group is that in? Right? The moth is in the same group as the bees and the ants. You can use the fancy term, or you can use the common term, whatever you want. And how many legs do they have? How many pairs? This is 4 pair. Okay. Crayfish, how many pairs? Insects, how many pair? That's the important term. How many pairs of legs? You guys can fill that out. Okay. All right, so here, together, the head and thorax are called the cephalothorax. The cephalothorax covered by the carapace. I explained to you what that was. Has specialization of segments occurred? Oh, yes, absolutely. Why? Because you have walking legs, right? Walking legs and gills in the thorax, in the cephalothorax. And you can give the example of they have the sensory organs like 2 pairs of antennae and eyes on the head also, and reproductive organs by the abdomen, etc. Locate the compound eyes composed of many individual units for sight. Do crayfish demonstrate cephalization? Oh yes. We have a head with eyes, plus antennae. Okay, don't worry about this one. We can't do that. They already asked this question, so we'll do that. External anatomy of the grasshopper. The hind leg is well developed and used for jumping. How many pairs of legs are there? You guys should know the answer to that. Is located in grasshopper adapted to land? Why? Because, well, they have wings, right? Explain. They have wings and jumping legs. You can't use those things underwater. What good are legs that are good for jumping underwater, right? Not very good at all. Okay, locate the spiracles, which I showed you. These openings allow air to enter the trachea air tubes, which constitute the respiratory system. Trachea take oxygen directly to the leg and wing muscles. Why is it beneficial for the oxygen to be taken directly to those organs? Those are the organs. Those are the organs that demand the most oxygen. Okay. We're not going to do the internal anatomy. Don't worry about this. Don't worry about these parts. I apologize. Okay, so don't worry about these mouth parts. Don't worry, we're not going to test you on this. Okay. Do know this, and like I said over here, to be able to identify the antennae, compound eye, the spiracles, the ovipositor, forewing and hindwing, and how much head, thorax, and abdomen separation of the body parts there, and be able to know 1, 2, and of course 3, right? Legs. And we don't have to worry about this. Comparison of crayfish and grasshopper. Locomotion, crayfish. Legs plus swimming appendages. Grasshopper has wings and legs. You can write jumping legs. Respiration, gills. Grasshopper, spiracles. nervous system. They're about the same for each. There's no difference. You can say, right, no difference. Reproductive features. Well, reproduction here is external, right? I wouldn't say external. How would I put this? You know, we didn't really discuss it, so don't worry about it. And there's no difference in sense organs, right? They both have antennae. They both have compound eyes. Let's say antennae plus compound eyes in both. Excuse me. Other than those shown in figure 16.14, name a type of insect that undergoes complete metamorphosis. Oh, I gave you a few, right? Bees, wasps, ants, excuse me, beetles, flies. How about incomplete metamorphosis? Well, you can have dragonflies. Right, they had the little predatory nymph that lives underwater, and then as it grows, it just adds little portions that look more adult as it grows, little by little. You got cockroaches, everybody's favorite insect. Yeah, dragonflies, cockroaches, other things like cicadas, those loud, noisy ones in the end of the summer. Okay. Ah, okay. The evolutionary tree of chordates shows that some chordates, notably the tunicates and lancelets, are not vertebrates. Okay, so those, here we go, lancelet, they look like little worms. This one looks like a squishy grape, this one. But they do have the other characteristics. They have the postanal tail, they have the notochord, dorsal nerve cord, and pharyngeal pouches. So that's why they're still chordates, these two. The adult version of this has those, is missing quite a few of those characteristics, but the larval stage has them. Okay. Those chordates retain a supporting rod called a notochord and are called the invertebrate chordates. Why? Because they don't have vertebrae. Missing the vertebrae. And remember, the vertebrae is the backbone. So they don't have those bones. You have jawless fishes, the agnathans, by the way. Like the lamprey, for example. This is a lamprey. You have the cartilaginous fishes, which are the sharks, the ray-finned or bony fish. These are bony fish too, lobe-finned fishes. And you have the amphibians, the reptiles, which includes the birds, and the mammals. Yeah, includes birds. There you go. Okay. The other animal groups in the figure above are vertebrates, in which the notochord has been replaced by the vertebral column. Vertebrates are segmented, as evidenced by the vertebral column. Fishes include three groups: the jawless fishes, the cartilaginous fishes, bony fishes. The bony fishes include the ray-finned fishes and the lobe-finned fishes. And what innovations, called out and figure, evolved among fishes? Let's see what they're talking about. Oh, yes. Jaws, bony fish, and lungs. Okay, so you got jaws, bones, and lungs for the lungfish. Okay. It's not quite the lobe-finned fishes, but the lungfish. And these evolved, this group evolved into the tetrapods, which are us, the ones that left the water. The terrestrial vertebrates are all tetrapods. There you go. Because they have 4 limbs. The limbs of tetrapods are jointed, right? Appendages, just like those of arthropods. Amphibians still return to the water to reproduce, but reptiles are fully adapted to life on land because they can produce an amniotic egg. The amniotic egg is because the embryo is surrounded by an amniotic membrane. Okay, let me draw that out for you. They don't have a picture of that. Here, I'm going to draw the amniotic egg. I'm going to try anyway, if I can remember. Okay, so here's embryo. Pouches and a little tail. Okay. All right. You have a yolk sac. And that's for food. You have another sac. Call it the allantois. That's for getting rid of waste. And then there's another pouch around that. And all right. If I remember correctly, there's the amnion, which is directly surrounding the embryo. That's the amniotic sac. And then you have the chorion. You don't have to know all of this. Yeah. Right, so that's the egg. So that's the eggshell. And you have the chorion, and then the amnion that surrounds the embryo, and the yolk sac, which feeds food to the embryo, and the allantois, which takes waste away from the embryo. That gets replaced. The yolk sac and the allantois eventually, in mammals like us, get replaced by the placenta. So yeah, you had a yolk sac and an allantois early on in your embryo stage, in our embryo stage, but that got replaced by the placenta. But the amniotic sac remains a fluid-filled sac, and um, oftentimes they do tests on the amniotic fluid of a baby before it's born to check for things like trisomy 21 or other problems with a risky procedure, and it's only taken when necessary. Okay. The amniotic egg is so named because the embryo is surrounded by an amniotic membrane that encloses the amniotic fluid. Therefore, amniotes develop in an aquatic environment of their own making, correct? Do all animals develop in water environment? Uh, do all animals? No, of course not. No. Insects don't need it. I don't think that's the answer they're looking for. I'm thinking, but whatever. We caught them. All right, so we have a little loophole of an answer. I think they're looking more for do all vertebrate animals develop in a watery environment? And then you could say yes, because the you can say yes because the eggs are either develop in an aquatic environment like a pond or the ocean, or they are amniotes, in which case they make their own water available for the embryo at in that isotonic solution. All right. Laboratory review. Name two types of organisms, which we didn't dissect, or we would not have dissected, that belong to each of these phyla. Mollusca. Ooh, that's a good one. Let's You guys can think of something, right? I can think of. I'll give you one. Oysters, right? You guys can think of another one. Annelids. I gave you two examples. Arthropods. All right, so I already gave it to you, so I might as well give it to you again. Earthworm and leeches. I kept giving you two examples. Arthropods. I gave you so many examples. You guys can do that. Echinodermata. Those are the sea stars. And we didn't talk about these, so I'll give it to you. Sea stars, and another one, you can say sea urchins, sand dollars, sea cucumbers, sea feathers, sea lilies. There's quite a few. Chordates. Oh, we discussed five or six classes of chordates. Okay, most of them vertebrates, so you can fill that in. Name the type of foot and location of the foot in a clam and squid. Okay, in a clam, it's tongue-like inside the shell. And in the squid is the tentacles. All right, so guys, make sure you find the answers to this and use the examples from the book. Because on the final exam for the lab, I'll be using those terms. So I'll probably just do multiple choice questions, but I'll use the animals that were given as examples here in the book. All right. Associate the type of foot in the clam and squid with their way of life. One is the filter feeder. That's right. That's fine. Just write this. One is a filter feeder, and the squid is a predator. And this is a question that was already asked. Both externally and internally, earthworms show evidence of segmentation. How? Okay, why are they asking? I don't remember seeing anything here about earthworms. Let me see this. Yeah, so I don't know why they're asking that question. All right. I think it's too much for you guys since we didn't discuss it. So let's eliminate it. Basically, they have little septa or little walls that separate the segments on the inside. On the outside, you can clearly see the segmentation because of the little constrictions between each segment. If I were to show you a picture of an earthworm, you'd be able to see it. Name one obvious adaptation of a crayfish and one adaptation of a grasshopper. Crayfish? Adaptation. It's just something that the crayfish has that no other animal that we've seen so far has. You can say gills, right? No other animal that we've seen in this chapter. While the grasshopper has those other two things, right? Spiracles. Did you say wings? Jumping legs? Okay. How do insects assist the transport of oxygen to flight muscles? They use trachea that directs the oxygen to the flight muscles. How do you know that the grasshopper? How do you know that? How do you know that the grasshopper adaptation you provided in question 5 is an adaptation to life on land? We spoke about spiracles and wings, right, or jumping legs. You can't use any of those. None of those would function underwater. What system is unique to echinoderms and what is its function? We didn't discuss echinoderms. We're talking about the water vascular system, so don't worry about that. What innovation during the course of evolution can you properly associate with these groups of vertebrates? Ray fin fishes. Bones. Sorry. Let me be more specific. Bones of calcium. Amphibians. I guess you can say metamorphosis, right? Metamorphosis for living on land. Or maybe they're just looking for lungs. Yeah, maybe just lungs. Reptiles. The amniotic egg. Amniotic egg. What type of skeleton both arthropod and vertebrates share? Is a jointed. And that's the last page. Okay, guys. So I hope you're still with me here, and I know it's a long chapter, but you guys can listen to it in pieces and parts. Not tomorrow, Monday. So guys, you guys have a good weekend. Monday we'll come back with another installation of this biology stuff, and we're going to start the dissection of the fetal pig. Okay, so the second lab exam will be, I do believe it will be, let me see. I haven't set a date yet. Okay, so the second lab exam will be after the pig dissection labs. And so Monday, Tuesday, Wednesday are the 3 pig dissection labs. Of course, we're not actually dissecting the pigs, but we're going to discuss the dissection of them. And we're ahead of the game by a day. And I did that on purpose because I wanted to give you guys an extra day to study and to prepare your lab manuals. This weekend, I will give you instructions on how to hand in the lab manuals and when to do so. There is a cutoff date. I need to have the lab manuals so I can give you a grade on them by a certain date. And that also includes the quizzes on the Plato to Darwin to DNA book. All right, so I know that I opened the quizzes until June 30th, but I'm going to have to cut that back because I need to have those quiz grades before a certain date. And I asked Dr. Muhlbauer what that date was. As soon as I hear back from her, I'll let you guys know. And The second lab exam is going to be a little bit different from the first, all right, because it's more of an identification test. You saw today in this recitation that I'm showing you of animals, and the pig is no different. All right, so that's actually 3 pig labs, and this one, that's 4 labs that are mostly identification labs, identification of body parts, and identification of where the animals belong in terms of classification. That's today's lab. So the second one, the second exam, will also be different because I'm not going to make it fill in. I find that it was a little harder than I expected. There's just so many ways for people to say the same answer. And that work of imagination on my part. So I'm going to make it matching or multiple choice, true or false. So that way there's no ambiguity in terms of answers. And you'll have a better idea what your true grade is immediately. Okay. So the second exam is going to be multiple choice, true or false, which is only 2 choices, or matching, which is a weird form of choice. But it should be easier because you're either right or you're wrong, and there's no way that you can misspell it or spell it the wrong way. The exam would recognize it as correct. Okay, so there's going to be visual identifications, more pictures, and so you're going to be asked to identify. I can ask you to identify the muscular foot, or the gills, or the mantle of a lamb, and the gills of a fish, or any of the things we went over today. The tentacles of the squid, okay, spiracle of the grasshopper, and all the little things we're going to cover with the pig lab as well. So it's not going to get. I can't say it's getting easier. To get a little bit harder because it's going to be a little bit different. So I'm glad that most people did really well on the first exam. Everyone did well enough on the first exam, but most people did really well. So that's good, and that'll hopefully offset the increased difficulty of the second exam. Okay. So again, I'll also have questions that are going to be based off the lab reviews at the end of the labs. But included with that, there will also be identifications. All right, and you guys, again, feel free to email me with any questions that you may have, and I'll try to get out instructions on how to prepare the lab manuals, and for the final submission. But you still have more than a week left before you have to hand them in. Okay, I think the last official day of labs will be not this coming Monday, but the Monday after that. And I think that'll probably be the last date that you guys can, maybe that Tuesday will be the day that you have to hand stuff in. All right. Okay, so have a good weekend, guys. Enjoy your weekend. Socially distant, of course. And stay healthy and stay safe.